So dinoflagellates are a phylum of algae that you might have heard about before, and they show a huge amount of diversity both in their lifestyle and in their morphology. Now you can see some dinoflagellates, like these three here, have these really well-developed armor plates made of cellulose that are known as thecae. Other dinoflagellates, like this Warnoweid here, don't have these thecal plates and are therefore known as athecates, but in ways that I'll describe later, these cells are still very far from defenseless. Now, in the same way that they show a lot of structural diversity, dinoflagellates show a lot of metabolic diversity. Roughly 50% of dinoflagellates are known as mixotrophs because they're able to carry out photosynthesis, and so they're autotrophic to some degree, but they still rely on heterotrophy and hunting in order to get enough energy and enough carbon to sustain life. Now, one feature that's common to all of the photosynthetic dinoflagellates is this triple membrane organelle called a peridinin plastid. Peridinin refers to the pigment that's unique to dinoflagellates. This pigment absorbs light in the blue-green range, which chlorophyll doesn't, and so this peridinin is able to complement chlorophyll in harvesting energy from light. Additionally, peridinin forms what are known as peridinin chlorophyll protein complexes, or PCP complexes, that are actually soluble. And so these PCP complexes not only utilize different wavelengths of light, but they actually perform photosynthesis in the cytosol as opposed to being bound in membranes like chlorophyll typically is in chloroplasts. Now, another difference between these triple membrane peridinin plastids and most photosynthetic organelles like chloroplasts is that chloroplasts only have two membranes. Now, the reason chloroplasts have two membranes is that they're a result of something called endocytosis, or actually more particularly endosymbiosis. Now, endocytosis in general is shown in these three images here, and it's the process whereby the plasma membrane or the cell membrane of some organism can form an invagination that surrounds some foreign particle or free-living organism, and then this invagination completely surrounds this external body and eventually pinches off and separates completely from this plasma membrane. And so if this red cell were to endocytose this green cell, you would see this third membrane that's derived from this plasma membrane separating the green cell from the cytoplasm of the red cell. Now, if this green cell were to survive this endocytosis, and if the red cell were able to somehow benefit from the survival of the green cell, this might develop into a permanent and stable endosymbiosis. And this is exactly the case with chloroplasts and mitochondria. Oh, I don't know what's going on there. All right. So chloroplasts and mitochondria are said to result from primary endocytosis or primary endosymbiosis. Now, dinoflagellates and a whole bunch of other algae actually resulted from a secondary endosymbiosis. And so this primary endosymbiont was endocytosed by a third free-living cell, and this results in a new organelle distinct from chloroplasts that actually have four membranes. And so this dark gray membrane is derived from the plasma membrane during the course of secondary endocytosis. This red membrane is the plasma membrane of this endosymbiont, hollow endosymbiont. This dark red membrane is actually derived from this red plasma membrane, and this final green membrane is the cell membrane of a previously free-living cell. Now, you'll notice here that there are actually four membranes in this organelle, but dinoflagellates only have three membranes. There we go. The most widely accepted theory for why dinoflagellates only have three membranes instead of the four that you would expect is that they had kind of a convoluted evolutionary path from where they were to where they ended up. And somewhere along that evolutionary path, they actually lost one of four membranes. There's an alternative theory that something called mycocytosis was used, which actually allowed this free-living cell to take up this entire cell without adding an extra membrane that results from this invagination, but that has yet to be demonstrated as true.